Morning. My name is John Wathen, and I am pleased to be presenting to EC 22 today. Most people in our line of work are aware that PFOS is the #1 actor among PFAS in U.S. waters, but this talk considers chemicals of this family in fish. There are chemicals other than PFOS in fish, but they tend to get lost in the concerns related to PFOS and water. This shot is of the Clifty Creek Power Plant on the Indiana side of the Ohio, just below Madison. This picture is of another of my favorite rivers, identifies me, and contains the all-important EPA U.S. government disclaimer. The balls on the wires are associated with a small airfield behind the trees on the right, and the west fork of the Susquehanna goes up to the left behind the camera plane. The next slide lists the 10 points of this talk. I'm not going to read them all, but I'm going to give each of them a name which is consistent with the content of this talk. Here are the 10 points of this talk. All over the country. PFAS in fish is entirely different than in water. Solubility. What is in fish is not in water, usually. Urban, non-urban. Repeatable patterns. Ubiquitous. Correlation. Variation. And RFDs are needed. This analysis is of PFAS data from the 2013-2014 National Rivers and Streams Assessment. Here's how you can learn all about the NRSA, get the data, and do your own analysis if you like. If I spent any more time on this slide, we'd never get through my presentation, but I do have to give advance credit to my colleagues and the organization that produced the data. Sincere appreciation to them for all this work, and I will name them at the end. These dots all represent sampling sites selected from the National Hydrography Dataset for rivers 5th order and above. Composite samples were collected and analyzed for total mercury, PCBs at 223 locations, and what this presentation is about, 13 PFAS compounds, or 349 samples. Since PFAS were detected in 348 of the samples, I can state with some authority that PFAS occur all over the country if there was any doubt before now. There are thousands of PFAS intentional and accidental products. Most of them are soluble as compared to, for instance, PCBs, but only a few occur in fish. When I saw this graphic in 2018 at this conference, I thought we'd never get a handle on all the PFAS. But what I'm going to try and make sure you comprehend now is that PFAS occurrence in fish is entirely different than in water. And that is more or less what this talk is about. In this study, 322 samples contained PFOS and variable concentrations of these other 5 compounds, PFUnA, PFDA, PFDoA, PFNA, and PFOSA, which occurred with significant frequency, that is, greater than 20%. 7 other compounds were detected with lower frequency, and 2 additional compounds, PFTRA and PFTEA, longer chain variants, were likely present but were not determined in this survey. I promise not to bore you with statistics, and this presentation is not about PFOS. It's about what else is in the samples. This slide was designed for you to scan and absorb the content because we have to get some basic numbers straight. Please note the 322 samples with other PFAS as well as PFOS with a mean concentration of just over 16 nanograms per gram PFOS, and the other 24 samples without other PFAS with a concentration of about 12 nanograms per gram. 322 is the denominator for co-occurrence of both PFOS and other PFAS. Now you can start to pay attention. These compounds are what this presentation is about. Please note their relative numbers and the percentage of samples in which they occurred. These are the PFAS that occur in significant numbers with PFOS, and I hope you want to know why. Also, please appreciate that PFOS is the only PFAS compound found in fish for which there is an EPA reference dose. 28 of all samples contain PFOS at a concentration greater than 46 nanograms per gram. It's all about PFAS solubility and the effect of chain length. Short chain PFAS are highly soluble. These diagrams, that I clearly did not create, illustrate that the solubility of PFAS decreases logarithmically with increasing carbon chain length. Long chain PFAS end up in fish because they are not soluble in water. There is also a length size at which they are too big to accumulate. The orange melons approximate what is possible. Here's some more thoughts on solubility and bioconcentration, basically here to plug some great sources of information. Derek Muir, co-author of the Martin paper, and Jason Conder. I must have read the Conder 2008 paper 10 times. The frame on the left depicts a lab experiment on bioconcentration and chain length, and Jason Conder explains that shorter PFAS chains don't bioaccumulate. I believe the folks at New Jersey DEP nailed the concept of long and short chain PFAS solubility. The frame on the left depicts no long chain PFAS at all in the water column, while the fish in all 11 of these water bodies contained robust concentrations of our three familiar actors. Also note that PFOS is right at the point where it is fully soluble and present in the water column, yet is totally at home in the organism, where there seems to be plenty of it. This slide shows us the water column concentrations for all the compounds that are largely absent from fish, except PFOS, as well as the total absence of long-chain PFAS from the water column, where we know they're not. Why? Because they're almost totally insoluble in water, and all of the mass is in the fish. The standing room only box describes a good part of the reason why PFAS with shorter chains are giving us fits. They are much more soluble than the other persistent organic pollutants we are used to dealing with, such as PCBs. And to summarize these few slides, except for PFOS, what is in fish is not in water, and what is in water is usually not in fish. This slide is almost an aside because when we sampled for PFAS in fish for the first time in 2008 and 2009, we were limited to urban sites because of resource constraints. This shows that except for PFNA, PFAS compounds occur at higher concentrations in fish from river sites in developed areas, suggesting an association between human industrial activities, waste disposal, and transportation with PFAS concentrations in fish. This is not true for all contaminants. Okay, now let's change gears and stop reading about solubility and such and begin to develop some terminology to knit this information together. SUDO is kind of an irregular acronym made up of the distinctive letters instead of the first letters of PFOS, PFUnA, PFDA, PFDoA equals SUDO, occurring together in fish as they usually do. And of course, this is anything but official EPA terminology. It's just a tool I have devised for this analysis and presentation to keep my tongue from getting too twisted up when I'm talking about PFAS in fish. 72 out of 322 of the samples look just like this, pseudo-4 and no other PFOS in the sample. And then there are pseudo variants, 76 out of 322, another 24%, more pseudo compounds with a variety of other PFAS. The simplest example is a single additional compound, such as PFOA, one of the rarer PFAS, rare in freshwater fish. Call this example SUDOC, PFOA being an original C8 compound. There are three additional recurring subgroups of compounds found among the samples that appear to invite further exploration. 29% of the samples include PFNA. That makes them SUDON. That's 93 of them. That's almost a third of the samples. And 14% of the samples include PFOSA, about a seventh, without PFNA. That makes them SUDOA. And with the descriptive listing starting to get a little cluttered, if a sample contains both PFNA and PFOSA, you have SUDONA. That's 11% of the samples. Not an upscale town in Arizona, that's SEDONA. What I hope I have illustrated here is that point number 6 is that on a large scale, PFAS in fish occur in repeatable patterns. Here's a summary slide for this pattern, which is what this talk promised. The SUDO compounds occur with high frequency. PFNA and PFOSA, and perhaps PFTRA, occur at a medium frequency. Welcome to Sedona. Other PFAS occur less frequently or at lower concentrations. I hope the three different groups are clearly depicted on this slide. This pattern of occurrence was not discovered in this analysis. I guess I am disclaiming coming up with anything new, and here I have to credit Washington and New Jersey certainly, and probably others, again for other than the nomenclature. But I do suggest that this is the first time that this pattern of distribution of PFAS compounds has been described across the entire country, and no one else knew what to call them. In aggregate, these same patterns are found in major rivers everywhere in the country. No relationship between the background concentration of PFOS and the relative amounts of any other PFAS is apparent, although I think we plan to do more analysis of that on the next go-around. I think it's worth noting that PFOS and the carboxylate PFUnA in fish tissue are not correlated to any material extent in these samples from major rivers. In contrast, the carboxylate PFAS, PFUnA, and PFDoA are moderately correlated at the R² equals 0.57 level that may reflect structural, chemical, and potentially source commonality. Digging a little deeper into this, PFAS molecules may appear similar in the basic PFAS structure, but the number of pair and polyfluorinated carbons, the nature of the functional groups, linear versus branched forms, etc., are associated with a range of dissimilar properties, solubility, toxicity, health effects, and other factors. Of the several compounds I have discussed today, there is only one with an EPA reference dose to its name, which is PFOS. You might be off the mark trying to figure out the toxicity of what is actually in a fish on a plate and be left fumbling with shortcuts. My favorite shortcut, one devised by folks in the Netherlands National Institute for Public Health and the Environment, uses values for multiple compounds as potency factors and might describe a less palatable meal on your plate than desired. Point number 10 is we need RFDs for all these compounds in order to assess the potential health effects that they are imparting to our fish consumers. Down to the last couple of slides, again showing the 241 sites with all four primary SUDO compounds. I do want to make well-deserved acknowledgments. Leanne Stahl and John Healy in the EPA Office of Water, where I also sit, Tony Olson in EPA ORD, Blaine Snyder of Tetra Tech, Harry McCarty with GDIT, and many others. And thank you very much for your attention. And I did not catch any of these fish.